This video presentation will correspond with your Chapter 9 handout. I've decided to present Chapter 9 in a series of 3 to 4 videos, so you don't have to listen to the entire thing all at once. So certainly you can come back and find whatever information you need or re-listen to, or whatever however you want to go about this. This first video that we're going to present here regarding Chapter 9, some of it will be familiar to the central nervous system lab, and so some of those sections that we've already covered in lab I might kind of walk through a little bit more quickly. So if we look at the nervous tissue, there really are two types of tissues that make up the central nervous system, the brain and the spinal cord. And those two types of cells that you'll find in the nervous system include neurons, which we've talked about a whole heck of a lot between Chapter 5, 7, and 8. But there are also cells that are there to help out the nerve cells do their job, and those are called glial cells, and they have an accessory role to the brain. If we look at the glial cells, we can properly call them neuroglia, and they're there to, as I mentioned, help out, and I have air quotes around the word support. We'll learn that's physical and metabolically, physiologically support the nerve cells. They do give physical insulation. They do aid in early nerve cell growth, like for example when a little baby is growing as a fetus in its mother's uterus. We have neuroglia that help make sure the nerve cells go to the correct, say, frontal lobe, parietal lobe, wherever the case may be. They're there for metabolic needs. The key thing to understand about the glial cells is they're not nerve cells. They're there to help, but they're not there to generate action potentials, and so they're not in the communication between nerve cells, but they're there to help nerve cells just survive and do their job. There are 5 types of glial cells. They're called astrocytes, oligodendrocytes, microglia, ependymal cells, and Schwann cells. You'll notice in this list that I have an asterisk by the Schwann cells, and this is because the Schwann cells are the only peripheral nervous system glial cells. So the first 4, the astrocytes, oligodendrocytes, microglia, and ependymal cells, those are found exclusively to the central nervous system, while the Schwann cells are found exclusively in the peripheral nervous system. Looking at what each does or is responsible for, the astrocytes, they get their name because they appear star-like, and under a light microscope they're kind of difficult to really kind of see that. Maybe someone used the word star-like to kind of describe them, but they do have these processes, as you can kind of see in this image here. The beigey colored cell is an astrocyte, and the nerve cell would be in the background with the yellow, and capillaries in the kind of pink colored. And the astrocytes are actually the most numerous glial cells that we have in the body, and as you can see, the second bullet illustrates, and as well as the image, is that they physically touch the nerve cells and associate with the capillaries. And the point is to not only physically support the nerve cells, but to bring those nerve cells close to their nutrient source, or the capillaries. It is the astrocytes that help young nerve cells grow into the correct region of the brain, and they also are critical for this thing called a blood-brain barrier. Some of you may have heard of this thing called a blood-brain barrier before, and effectively what it is the protective mechanism that your body has to ensure that pathogens from the blood don't get into the brain, so to prevent brain infections, basically. So the astrocytes help in establishing that blood-brain barrier. They're also involved with repairing brain injuries and getting rid of, waste, if you will. If their tissue's been damaged, they can kind of clear out the interstitial fluid and make sure nutrients and repair factors are kind of brought into the area. Astrocytes will also get rid of any excessive neurotransmitters so that there's not unnecessary messaging. So they're kind of cleaning up, bringing in nutrients. In addition to getting rid of excessive neurotransmitter, the last item shown here is that they do get rid of excessive potassium from the extracellular fluid. You probably have an idea that ion concentration is critical to nerve cell function, and so the astrocytes can help make sure that potassium levels are balanced in the extracellular fluid. Another glial cell are called microglia, and they get their term because they're small. And you notice they have the same kind of processing look, but they're much smaller when contrasted to the astrocytes. The primary job of the microglia is to have phagocytic function. And if you remember that term from chapter 4, I believe it was, the process of phagocytosis is to engulf large foreign particles, and so that's what the microglia do. They give immunity to the central nervous system. Another type of glial cell are called ependymal cells, and those cells are the cells shown here in the green. And you'll notice maybe that the ependymal cells have these ciliated projections. If you remember from your kind of general biology classes, the role of cilia are to either move an entire cell or move fluid across a cell. Here you can see that the ependymal cells are actually anchored together, and so their job is to actually circulate cerebral fluid across them. So they keep kind of fluid flowing within the central nervous system. My abbreviation here, CSF, stands for cerebral spinal fluid. As they line the cavities of the brain and the spinal cord, keeping that cerebral spinal fluid moving. Fourth type of central nervous system glial cell is called oligodendrocytes, and we've actually covered these, discussed these in chapter 7, and so I won't address them much here, other than it is the oligodendrocytes that provide the source of myelin for our brain and spinal cord, and we know the role of that is to enhance the rate of conduction within a nerve cell. If we look at the nervous system in general, our nervous system is going to allow for homeostasis, in that it provides the ability for the body to detect different modes of stimuli, whether they be mechanical, recalling from lab, mechanical, thermal, chemical, and electrical. Whatever the mode of stimulus, our nervous system can acknowledge those. You might be more familiar with the nervous system in its role of integration or association, or kind of controlling different processes, but it can also coordinate motor output. If we look at the central nervous system more in detail, we'll see that the cell types in the central nervous system are both gray and white matter. What that means to you is gray matter, some cells are not myelinated, and white matter means that some cells are myelinated. Size-wise, the brain is about 3 to 4 pounds, which isn't very large. But if you think about how well it's designed with all the folds in the cell, you can get upwards of 100 billion neurons in an adult brain. But it receives a disproportionate amount of blood flow, right? 20% of the body's blood flow is continuously going to the brain. And what you'll see is 3 to 4 pounds is not 3 to 4% of the body. So this tiny little structure is receiving a 1/5 of the total blood flow. In chapter, I think 15 maybe, we'll give this organs like the brain who receive this disproportionate amount of blood flow called reconditioning organs. Okay, so the brain is going to receive a lot of blood flow. This is because it needs a constant input of oxygen and glucose. I have in parentheses some values. The brain can go no more than 4 to 5 minutes without oxygen before you begin to see some cell death. And then glucose, so if the brain goes about 10 to 15 minutes without glucose, the brain cells will begin to die. So that backs up why there's such a high proportion of blood flow going to the brain alone. If we look at why this is, the brain relies upon aerobic respiration of starting out with glucose, so starting out with glycolysis, all the same steps that we talked about before, the intermediate step, Krebs cycle, electron transport chain, which we know requires glucose and oxygen to take place. Furthermore, the brain and the rest of the central nervous system does not have the ability to store glycogen, which is a storage molecule full of glucose. And fatty acids aren't used. In extreme conditions, ketones can be catabolized for nutrients, but the brain prefers oxygen and glucose on a regular basis. The next component that we're going to discuss here, some of it will be a review from the Central Nervous System lab. So if you have any further questions, obviously you can email me or you can refer to your Central Nervous System lab. So we're first going to look at the outer structures of the cerebral cortex, including the lobes, the frontal lobe, parietal lobe, temporal lobe, and occipital lobe. The cerebral cortex is alternatively called the cerebrum, and that's the biggest part of the brain. So about 80% of that 3 to 4 pounds is the cerebral cortex. And this is what allows for us to have what's described as higher mental functions, right? So your thoughts and emotions and planning and all those other fun things that makes us uniquely human. As discussed in lab, the brain, you have one brain, but anatomically we have the left and the right hemispheres. And we also learned that the corpus callosum is the structure that connects the two hemispheres. Okay, so I'm going to try to highlight that. Okay. So the corpus callosum I'm going to try to highlight here in the blue. Okay. Is that band of white matter that physically connects the left and the right hemispheres. You may recall from your central nervous system lab that had you guys looking up primary areas and association areas. And what that is different regions of the cerebral cortex are dedicated to different functions. And those areas that help coordinate motor output are appropriately called motor areas. Those areas that allow for sensation perception and awareness include what are called sensory areas. Again, whether the sensation be physical, like what we saw in the parietal lobe, or visual, like we saw in the occipital lobe, or auditory in the temporal lobe, those sensations are processed in areas of the cortex called sensory areas. The rest of the brain, the cerebral cortex, that did not directly coordinate motor output, nor directly process some form of sensation, those are just called association areas. And if you remember from your drawing, that was like a lot of the cerebral cortex is just association areas. And what they do are like the control center where they compare, and the term I have here is integrate. But they take in multiple modes of information and kind of try to make sense of incoming information. Figure 911 A and B illustrate a couple of views of the brain. The image on the left is looking at what's described as the anterior or the front of the brain. You can see the folded cerebral cortex up here on the top portion of the brain. You can actually see a nice little view, anterior frontal view of the brainstem, and then hanging on the back you can kind of see the cerebellum. In the image on the right hand side, we can still see the same structures: the cerebral cortex, the brainstem, and the cerebellum. You may recall as well from lab that we looked at the different lobes. The lobe that is described as in the front is appropriately called the frontal lobe. It is separated from a region that's called the central sulcus. And I tried to highlight this for you guys in your little brain dissections that we did last week, but it's that little slight division that you can see between the frontal and the parietal lobe. The parietal lobe would be posterior to that, and then the most posterior portion of the brain, highlighted in green here, is the occipital lobe, and on the far right, far side, highlighted in yellow in this image, is called the temporal lobe. For this section of class, the frontal lobe we learned, you remember in lab, we get our thought processes, our emotions, and our personality traits. But for this class, we're going to focus on an area of the frontal lobe, this thin strip that's red in the image, and I'm drawing a blue highlight through. It's called the primary motor cortex. The primary motor cortex is that part of the brain that allows for us to voluntarily move specific regions of the body. The way it's arranged, you can see a similar image, figure 9.15a, but the way that the gray matter in the primary motor cortex is so that however much area of the brain is necessary to move a particular part of the body, right? Let me restate this maybe better. If more precise motor control is necessary to move an area of the body, it's going to get more area of the brain that controls it. Okay, so for example, if you look in the image, you'll see that the brain looks really huge, or not the brain, excuse me, the face looks really disproportionately huge. I'm not saying that the face is huge. What it's saying is it takes a lot of gray matter to control all the muscles in the face. And in your handout, there's an area that says the area which it controls is something depicted in. And what it's depicted in is this thing called a motor homunculus, okay, highlighting in blue. What that means is a little monster. Do you remember in the very first 1 or 2 lectures that we had, I showed a video of the human body, and it had a little claymation sculpture of a person that had huge hands and huge lips. Okay? That is a homunculus. And what this is telling us, or trying to illustrate, is that's how our brain kind of perceives our body. And so when you see a lot of area in the frontal lobe controlling the face, because the face is really important. If you contrast that to the toes, and not so much gray area. Gray matter is controlling the toes or the ankle or the knee. Okay, so it's called a motor homunculus, illustrating the control from the frontal lobe and that primary motor cortex. Behind it, behind the frontal lobe, at that central sulcus, is the parietal lobe, and we also talked about it in lab. The parietal lobe is shown here in blue, and again, change colors. It too had a lot of gray area, but we're going to focus only on this little strip of gray matter in this. It's called the primary somatosensory cortex. In lab, your book called it the sensory cortex, but I'd like for you to add the prefix on there, somato. I'm going to highlight this back to blue. Somatosensory cortex. And if you remember, what does the word somato mean? Okay. Somato or soma means body. Okay, so it's body sensations. It's part of the brain that regulates body sensations, and these are physical sensations. It too has a similar homunculus. Okay, so its depiction is in a sensory homunculus, and you'll notice that the face is still disproportionately large. When you think about it a little further, you know that you can really pick up sensations well with your face versus, say, the bottom of your toe, okay, or your knee, or whatever the case may be. Okay, so again, the area that the the parietal lobe monitors is depicted in a sensory homunculus. This next statement that I have here presented basically illustrates that your brain is not static. It's ever-changing, or the proper term is called, it's plastic. And so, depending on how much the brain is used, is going to dictate how much the brain can change and adapt to. Right? So, because not two people don't experience things the same, their brains would change differently, even though the scenarios may be the same. So our brains are very much plastic or moldable, or considered to be plastic. Okay. I'm going to go ahead and conclude this video here, and you'll look for another video called or titled Chapter 9A.